So it's a really great pleasure to welcome Alison Banham from Oxford. We couldn't really think of a better person to, always the perfect person to start this meeting.
Oh, thank you very much to Andy and the other organisers for inviting me to come and talk to you today. So I've been making mouse monoclonal antibodies for over 25 years, both for our research and also as a facility for some of our grant holders in the past. What I'd like to start off with is to introduce you to the European Monoclonal Antibody Network, which is something that I've been a founder member of. So we started out really because one of our lab went off to Madrid to set up her own group and wanted to actually have a way of talking to other people that were still making antibodies. So we've gradually expanded the network. We now have 18 laboratories from 10 different countries, and we try and meet sort of annually to share our experiences. So this just gives you a look at our homepage. And the idea is that this is largely academic labs that produce and characterize their own antibodies. So what we want to do is to be able to share knowledge. So we have people doing all sorts of different formats, including nanobodies. We want to improve not only our own technology, but also find ways of standardizing and improving what we do sort of across the board. We have our own catalogue of antibodies. So you can see here we have a catalogue of human antibodies and antibodies to mouse proteins, and each of those has a proper validation file showing how we've said that it's fit for purpose in the various applications we recommend it for. We have some standardized protocols that are available to anyone that wants them. We also have a private area so we can share things perhaps that aren't published, just among the members as well. Certainly in this audience, this isn't a surprise to anybody, that basically if antibodies aren't well validated, we just spend so much time and money trying to sort it out, and also trying to deal with data in the literature which just isn't appropriately controlled. So what we wanted to try and do in EuroMabNet was come up with something that looked a little less like my daughter's bedroom, and really something a little bit more organised, because there are so many antibodies available from so many different suppliers. How can somebody who's fairly new to antibody research go out there and choose an antibody that's going to work from them? I think we decided we didn't want to be the antibody police and say, these are the rules, this is what you must do. What we wanted to do is to provide guidelines saying, if you want to find an antibody, this is how you can find it. This is how you can decide whether it needs validation, and really just provide people with practical tools to know what they ought to perhaps be able to do. So we published that paper earlier this year in the journal MABS, and it was a sort of really a researcher's guide to finding and using antibodies. So on the web, we actually have clickable links. So we have several steps, one of which is obvious, you need to know what your target antigen is. It's surprisingly not always obvious to people. I have had people where they thought they were looking for one thing and actually it was called something else. You need to know what applications you want it for. And then really finding out if antibodies are already available and not perhaps picking up the only antibody catalogue you have in the lab, but understanding that there are lots of tools on the web like BioCompare and ways like CiteAb of finding out which are used already in the literature. And by the time you've got all that information, you have to decide, can you just use that antibody? Is it well validated? Everybody knows that. Or do you have to do more validation? And then to give people ways of explaining how you validate antibodies. And also, you must publish what antibody you used, where you got it from, and you have to have reproducible results and share them with the research community, because otherwise there's just no point. I'm not expecting you to be able to read all of that, but this is just to say if you click on something, say which is reactivity with the target antigen, then there's an awful lot more information available about showing it reacts with perhaps the immunogen, with a recombinant antigen. That, of course, doesn't guarantee it will recognise the endogenous antigen. Even that doesn't guarantee that it'll be specific for the endogenous antigen. Then there's things like, does it do what you expect in terms of what else is known about it, what you know about transcripts, and also some of the confidence you can get if you're actually working with more than one antibody that perhaps recognises different epitopes. Often that can really help give you confidence. And then each of these has worked examples showing how, for a particular project, all of those various steps worked through, so people can see what we were doing. And this again is something where we had antibodies to PIM2. Now this is transfectants that are epitope tagged. These are PIM1 transfectants. We know they're expressed, they've got the V5 tag, no antibody binding. Again, in ELISA, the antibody recognised PIM2, not the other two family members, and taking it through onto paraffin-embedded cell pellets, again, the PIM2 transfectant stain, and not the other two PIM family members. So it just gives people a chance to actually look through a lot of data and see how it worked. There have been some really interesting surveys recently, and this survey by the Global Biological Standards Institute really highlighted that the more experience you have of antibody validation, the more you understand that it needs to be done. And I think the fact that 30% of young people who'd done antibody research for sort of less than 5 years didn't perform any antibody validation, and a quarter of them thought it was unnecessary, is really quite worrying. So I think one of the things we also have to think about as a community is not just how we validate things, but actually how we educate younger scientists and train them to actually use antibodies and their validation. Now, we can't tackle that problem all at once, but one thing that EuroMabNet's doing for the first time this year is we've actually hosting a small workshop where we've got about 40 PhD students who are going to come and learn about antibody validation. They have a chance to submit problems that they've got with validation and ask for help. And also we're going to talk to them about some of our experiences and share those. Certainly I'm going to be talking to them about what happens when it isn't validating. How do you go back? Do you ask for your money back? If you find it has an autospecificity, can you persuade people to change the data sheet? And what happens if somebody's published something and it's wrong? What reception might you get? And it can be quite varied. So basically that's all I really wanted to say about EuroMabNet. And now I'd like to talk to you about some validation that we're doing in my own lab with an interesting class of antibodies. So this is a class of antibodies that are targeting intracellular molecules. And we call them Tcell receptor mimic antibodies because they bind in the way that Tcell receptors do. And they present a particular challenge for validating their specificity. Everybody knows that therapeutic antibodies are some of the most effective drugs, particularly in the cancer arena. But largely the targets have been either cell surface or secreted proteins because with big molecules you can't get them into cells. And certainly a few years ago you can see a very small number of intracellular molecules being targeted. But when you look at the proteome, it's quite clear that there's an enormous repertoire of molecules inside of cells that make exciting targets. Now, the immune system has already worked this out for itself. So you can see here that you have your protein, in this case the unfortunate chicken, which is processed by the proteasome, which dices it into peptides, or in this case the drumstick. This then goes into the endoplasmic reticulum where it binds a protein called MHC class I. Those peptides are then presented by MHC class I on the cell surface. And this is how T cells conduct immune surveillance. So we have a way of targeting intracellular antigens through these cell surface presented peptides. Now, there's been a lot of historical targeting of these. People have used vaccines to try and boost the amount of peptide presented to try and get a better immune response. There's a lot of interest at the moment in using T-cell-based therapies, including things like these chimeric antigen receptor or CAR T cells. And the interesting thing with these antibodies is that they bind in a very similar way to the T-cell receptor. So normally you'd have an antigen-presenting cell, MHC class I and a peptide, which is then recognized by the T-cell receptor. A TCR mimic antibody is binding the same particular peptide that's presented by MHC class I. So what you actually have in terms of your target antigen is a dual molecule epitope. You're going to have an MHC class I allele, and we have quite a lot of those, so you have to choose one, and a 9 to 10 amino acid peptide that binds that, derived from your intracellular antigen. Now, what we decided to do was to target wildtype P53. Now, P53 is a fairly universal cancer target. It's mutated in over 11 million cancer patients, and it can be deregulated by other mechanisms as well. We decided not to target mutations because it would actually cut down the patient groups available greatly, although the specificity might be greater. And indeed, actually, when looking at the top priorities for cancer vaccines and immunotherapy, wild-type P53 was among the top 10 prioritised cancer antigens because it isn't widely expressed in normal tissues. And even the wild-type epitopes have enhanced presentation on tumour cells. So there's a window where you can get in and hopefully target tumour rather than normal. So we had to choose our peptides. So what we've done is we've chosen p53 peptides that we knew from vaccine studies were endogenously processed and presented on the cell surface. And we'd had vaccine trials and there'd been no adverse side effects using them. We chose a very common MHC class 1 allele, which I refer to as HLA-A2, which is present in roughly 50% of the population. And we chose our peptides in regions here, peptide 1 and peptide 2, that have very low mutation frequency because we obviously were going for wild-type peptides. And we also chose them upstream of the mutations that actually terminate translation because these sort of cut in around about here, and at that point you're not going to get epitopes if those patients have those mutations. So in terms of the immunogen, we make classical mouse monoclonals. So first of all, we had to make our HLA complex. So in bacteria, we express the HLA-A2 heavy chain and the light chain, which is beta-2 microglobulin. In vitro, we then refold those with our target peptide to make a monomer. That's then FPLC purified. See here the peak. On an SDS page, we want the band that actually is going to pick up both the heavy chain and the light chain. Those monomers are then biotinylated and created a tetramer with streptavidin, because that gives you much better binding and stability. And we actually had a peptide-specific T-cell line, so we could check that T-cells were able to see our p53 peptides presented in our tetramers, and you can see here that they did. Now, we started out with ELISA screening, and we had two different types of HLA monomers. We had one containing our p53 peptide and one containing a completely unrelated peptide from flu. That was to give us a chance to pick out all of those antibodies that just picked up the MHC class 1 backbone, and I'm not showing those here, and there were a lot of them. So this is the best of a very large screen. The antibody that we're particularly working with, I boxed in orange so you can follow it through subsequent slides. You can see here one where we just don't have enough specificity for the p53 containing monomer. It's also picking up the flu. We had some that looked great, and then when we cloned them, they had more background binding than was acceptable. And we had others that looked great, and then when we cloned them, we just lost reactivity altogether. So we ended up with 6 antibodies, 3 to each peptide. We knew that they bound our recombinant immunogen, but we wanted to see, can they bind a peptide presented on the cell surface? So we've taken advantage of an assay called a T2 presentation assay. Now, these cells are deficient in a transporter related to antigen processing. So they have HLA, but when you pulse it with the peptide, you can then basically direct a lot of the peptide you choose onto the cell surface. And then you can basically ask whether it then is stained by your antibody. You can see here we're getting nice shifts when we pulse with the p53 peptide. When we pulse with the flu peptide, it's overlaying our isotype control. We've got a little bit of binding on that one. So at that point, we're saying we at least do have some specificity for the p53 peptides. Only one of those 6 antibodies actually bound cancer cell lines. We're not entirely sure why, but with our specificity testing, we have some ideas, and we'll come back to that later on. But you can see we're getting binding to different tissue types: lung, colon, melanoma, lymphoma, and breast. And certainly at this point, it was about 12 of 34 cell lines that were positive. Now, we wanted to know, is that binding restricted to cell lines that have the right HLA haplotype and that have p53 protein expression? So here we've done Western blotting with 3 different p53 antibodies. We've done HLA-A2 staining by flow. And in this case, all of the cell lines stained by our antibody were HLA-A2 positive and had p53 expression. Now, we wanted to think, obviously, are we going to pick up normal tissue expression? So from the literature, one of the tissues that does express wild-type p53 are PBMCs. So we were able to look at those from 14 different patients that were HLA-A2 positive, and we only got slight staining on one of those buffy coat preparations. We actually then purified the antibody. We still saw that reactivity, but that particular patient had an enormous proliferation of granulocytes. So that granulocytosis could have been an indication of a health problem. So I think the feeling was, at that point, we weren't getting a lot of normal tissue staining. God, I'm going to choke to death now. Sorry about that. It's not contagious. So we wanted to know, okay, we've got these antibodies. Do they do anything in vivo at all? So in this first experiment, we've got a triple receptor negative breast cancer xenograft, and we're giving the antibody twice weekly at the same time as we give the tumour cells. And you can see we get a really significant delay in tumour growth with an antibody isotype that engages murine effector cells. We then wanted to say, okay, can we actually then let tumours grow until they're palpable and then try and treat established tumours, which is what we've done here. And again, we get a delay in tumour growth. Now, there's a cautionary tale about reagents, including Tcell receptors, and their slight promiscuity. So Immunocore had an affinity-enhanced Tcell receptor against MAGE-A3, which is a cancer target, and then they found that there was actually some cardiac toxicity, and the first 2 patients that got the CAR T cells died. And they actually came up with a very clever way of doing screening to try and identify what some of those cross-reactive toxicities might be, and was saying how important it is that you adapt that. So we thought, well, that should work also for our Tcell mimic antibodies. So what we've done is we've done individual amino acid substitutions all the way across our peptides with alanine and with glycine. We've then used that T2 presentation assay where we're putting the peptides on the cell surface. First of all, asking, can they bind HLA-A2? And pretty much most of them do reasonably well. And then, can our antibodies see those peptides? What you can see very clearly is that that arginine at position 1 is absolutely required. We lose our antibody binding if that's mutated. And in fact, what we did was we came up with a consensus whereby if you have to retain 50% binding, then that amino acid is required. So this is what we came up with here, and we were able to then search that through a database. So we came up with 20 different human peptides that shared that consensus motif. A lot of them were unable to bind MHC class 1. Some of them did, and some of them also bound by our antibody. Now, interestingly, of the 3 that give more than 50% of the binding activity we see with the p53 peptide, 2 of them have incredibly normal tissue-restricted expression, largely to immune-privileged sites like testis and brain, which we wouldn't expect our antibodies to be a problem in. And none of them have been described as T-cell epitopes in the literature, and that's including mass spec studies I trawled through databases. So we were thinking, well, that's just great. It's all looking fine. And then we stained more cell lines, which you do because you're still paranoid, because the idea this might be a therapy. You don't want to find out you're wrong by killing somebody. So we stained HL60, which is HLA-A2 negative, stains by our antibody, and in fact is also p53 negative. So at this point, we know that we've got some specificity issues. Now, interestingly, there was a Tcell mimic antibody against WT1, which was crystallized. And they actually found that this RMF motif was absolutely critical for binding, particularly that first position arginine, which actually you can see is very similar to what we're seeing with this in ours, where we have RMP. Also, they actually got slightly wider HLA binding. They stained multiple subtypes of HLA-A2 with that Tcell receptor antibody. So that's quite interesting too. So we had a poor PhD student that we sacrificed who basically did all of the individual amino acid substitutions one at a time. We're not doing the combinations because that's ridiculous. But what you can see here is that basically position 1 in black means that you can't substitute it, you lose your binding. It was absolutely required. Whereas other positions, like here, you can substitute all of the amino acids and not affect the antibody binding at all. So what we've done is we've come up with a consensus of what our antibody will tolerate in terms of those single substitutions. Now, we didn't search a sequence database because we knew what that would give us, very large numbers. What we've done is we've initially searched an immune epitope database, so experimentally determined MHC class 1 epitopes for this HLA-A2, and we came up with 300 epitopes. Interestingly, some of those were some of the very top cancer epitopes, such as NYSO1, WT1, the peptide recognised by the other T-cell receptor mimic antibody, tyrosinase. And in fact, when we tested these with our p53 antibody, they are indeed bound. So certainly it's able to recognise multiple tumour peptides. Now, obviously, that's only going to be any good if it doesn't recognise normal tissues. So what we've been doing at the moment is trying to get, first of all, an IHC assay up and running. So we can do that with our T2 pulse cells. We pulse them with p53, we get some staining. Pulse them with flu, we don't. With our cancer cell lines that are strongly positive by flow, we get staining. Some of those which are weakly positive, we can just see staining. At the moment, it's not perfect, but I think it's the next step for us is to try and do some IHC on normal tissue panels and see what staining patterns we get. So I think in summary, what I'd like to say is we've got this T-cell mimic antibody. It definitely recognises an awful lot more than p53. Whether it's ever going to be relevant for therapy, it'll be determined by us looking at a lot more normal tissues by IHC and also by flow, buying in suspension cell lines from a variety of different sources. We're going to have to do an awful lot of validation before we decide whether we can use it therapeutically, but it's still an interesting reagent in terms of studying antigen presentation. And one of the nice things about it is that it works extremely well for imaging. So we have a group in the Cancer Centre who have done radiolabelling of the antibody. And here we've got mice which have the breast xenograft that we've used in vivo. Our antibody labels that extremely nicely. You can see we don't get labelling of the tumour with the isotype control. So I think one of the things, if our screening of normal tissues looks positive in that we're not seeing large amounts of normal tissue staining, then I think we're wondering whether we could perhaps go in with a subtherapeutic dose and do some form of imaging to actually see where in people this antibody localises. So thank you very much for your attention. I'd like to thank Cancer Research UK, who funded this work, Demin Lee here, who's been absolutely critical for this particular project, and I'm very happy to take any questions that you might have.
Thanks very much. That was a great start. So we have got a roving mic, but I suspect people can probably just shout if you would like, or I can pass the mic. So we've got questions.
Extremely interesting presentation, very impressive. Did you also try to do mutations on the HLA molecule, amino acid substitutions to find out the contact sites?
We haven't. I think what we've decided we're going to do is, with the same student who's done that specificity screening, we're going to do a crystal structure. Because I think certainly from the ESC1, the WT1 antibody that was crystallised, that gave them those points in the HLA molecule that predicted that it would bind different HLA-A2 subtypes. So I'm wondering whether we've got something that's conserved in MHC class 1, but not the HLA-A2, but HLA-A1, which would be the HL60, and one of those peptides is able to be presented by HLA-1.
I was interested by the course. I mean, it's changing the topic completely, but the course you were talking about for training PhD students and postdocs things, it sounds like a really good idea that I hadn't sort of heard about before. I was wondering how successful it's been. I haven't run yet. But
well, we set it up fairly late.
Yeah.
But we did manage to get some sponsorship for some money to send people out. So yeah, we've got 40 coming.
Right. Okay. And those are from people within labs already in the network?
Some of them, and some of them not.
Okay.
Certainly some of them in the host country have come as well.
Yeah.
But I think certainly a lot of us speak about antibodies, and we get asked to talk about antibody validation quite a lot. Can you come and talk to people?
Yeah.
So.
Yeah, no, that's
I have one.
John.
John Collins from Virginia. So can I also ask about the training capability that perhaps your website provides? At the moment, it's best practice is illustrating. Have you got any educational functions developed on it yet?
No, we haven't.
I wonder whether that's the type of thing this community could do, that we could be putting up almost gamification of this training, that we could be putting up problems and just inviting people to...
I think sometimes it's, I've got this problem, is there somebody who can help me? And having dialogue, because sometimes labs work in isolation.
I didn't mean that's a problem, but I meant data handling problem. So. sort of so that again people get familiar with simulations of how we look at data, what are the flaws in it,
Yes.
sort of, what do we do next?
No, absolutely.
One more thing. I was so fascinated by this antibody. It's really so interesting and powerful. What I do not quite understand is if this antibody can recognize a lot of peptides from a lot of different proteins, and you don't see staining on normal leukocytes. I mean, normally it says you not have p53, but wouldn't you think that they should have any of all the other proteins that could be recognized?
Well, certainly for some of the cancer targets, I know they don't. So things like WT1, they don't. So I think it's going to be impossible for us to say what peptides from what antigens this antibody can recognise, because we've just done one amino acid change at a time. If you think about the combinations, so I think for us we're going to have to say we made it to p53. We know it can pick up multiple tumour antigens. And for us it's now, certainly it can't pick up ubiquitous antigens because it's not staining PBMCs. But maybe when we put it on a normal tissue array, we're going to get things like pancreas and lung coming up, and we'll say, no, it's not a therapeutic. But I think you've just got to start somewhere and go in a logical way.
Sorry, have you looked at the effect of any post-translation modifications on that arginine, and does that, if that's modified, does it affect the agonicity?
We haven't looked.
One front.
Immunocore.
So can you actually sort of run some proteomics on like a column with your antibody and precipitate like class 1 to look at what peptides?
Yeah, that's certainly something. I mean, we've certainly been doing MHC class 1 IPs just to check with things like our p53 targets. So the cell line that we actually used in vivo, we did IP the HLA-A2 peptides to make sure our target peptides were there. And although we picked up things like peptides from SHANK1 and bassoon, it wasn't the peptides that our antibody is predicted to bind. So we certainly know that those antigens can present peptides, but we didn't see those.
You and your network are academics who make antibodies. Do you think more academics should make antibodies? I mean, from scratch?
I think it's an investment in time and skill, and you need people who know what they're doing and learn.
Sort of dying out of it.
Well, I think there's so many reagents available.
Yeah.
I like the degree of control it gives us. I like the fact that we have panels, and I can choose myself what I think works best in our hands. And obviously we sell them back to the research community, and that really helps too. But I think that also puts us in both camps. So although I'm an academic and a user, equally I also understand some of the experiences that people have when they supply things. Okay.
Any more questions? Probably a perfect note to finish on, isn't it? Thanks very much.